Good afternoon, guys. Today is the last lectures of our course, and this lecture about microorganisms are causative agents of caries, infectious diseases of periodontium. But for initial, we talk in general about normal flora of human body. We talk about normal flora of the oral cavity, and then later we talk about bacteria, which may be present in oral cavity, sometimes constant, sometimes transitory, and can cause different type of diseases, which actual for stomatology. As I said, the first we talk about normal flora of human body. Maybe you all know that in some of biotopes of human organisms present normal flora bacteria, which very important for health of the body. And on this slide to show what biotope or what site of human organisms have these bacteria. For example, that is usually site which contact with environmental. That is, if we talk about nose and oral cavity, means mouth, then in nose, Haemophilus, Staphylococcus, Neisseria, Streptococcus. About mouth, only several groups, really it's a more. Fusobacterium, Staphylococcus, Streptococcus, fungi, Candida, Haemophilus, Veillonella, Actinomycetes. Then in gastrointestinal tract may be present Lactobacillus, Helicobacter, Streptococcus, Peptostreptococcus, Staphylococcus. It's in stomach. In small intestine, bifidobacteria enter big family, some of species and genus from family Enterobacteriaceae. It may be E. coli, etc. Clostridium, Enterococci, Bacteroides, Lactobacilli. And in colonna, E. coli, Proteus, Klebsiella, Pseudomonas, Enterococci, Acidobacter. In rectum, Bacteroids, Diphtheroids, Staphylococcus, Streptococcus. We bind in nose and mouth with gastrointestinal tract, but also we have urinary systems and reproductive systems which contact with environmental. That's why in urinary and vagina tracts present Candida, Gardnerella, Lactobacillus, Peptostreptococcus, Diphtheroids, Streptococcus and Bacteroids. Obligatory. If we talk about respiratory tract, lungs should be sterile, but until trachea may be present some of bacteria which can transfer through the mouth. And obligatory, we should be said about skin, because skin it's a protective layers of our organisms which contact with different type of bacteria from water, from environment, from air, and some of bacteria always present on the surface of the skin. For example, it's maybe Staphylococcus, Bacillus, Propionibacterium, Candida, Streptococcus, sometimes Diphtheroids. That's why, as you can see on this slide, a big amount of bacteria, big range of genus and species present in different site and biotope of human organisms. But some of site or biotope should be sterile because it's absent contact with environmental, and normally for health organism should be sterile. Nerve systems should be sterile. Blood systems, means heart, blood vessels, should be sterile. Liver, kidney, lungs, as yes, a low part of respiratory tract, high part of reproductive systems, and top part of reproductive systems and urinary system also should be sterile. Normally, atropine in healthy organism, bacteria absent. If it start to isolate bacteria from this site, as from blood, for example, that is pathology, that is diseases as sepsis. If it's isolate bacteria, for example, from cerebrospinal fluid, and you know about this, that is also pathology. It's not normal, it's diseases as maybe meningitis, encephalitis. Then, that's why we generally characterize site in which present normal flora of human body. And the next, what we should be discuss, why need normal flora? Why these bacteria for health organisms should be present on this site? Or maybe need that is all should be sterile and we should be treating and washing all of this site. Really, it's one of the factors of our immunity, one of the factors of our defines our organisms. Let's discuss what the main function of microflora. The first, it's called one of the factors of innate immunity, colonization's resistance of normal flora. What it means? It means that organisms which present in this site to prevent the colonizations of the body biotopes by foreign microorganisms, including pathogenic microbes. In other words, if it's on this biotope present normal flora, pathogenic microbes be very difficult to also sorptions and start to multiply in this place. But if this site be clear or means be free, then pathogens are more easy desorptions and start to invasive or maybe cause diseases. The first, colonization resistance. The next function of normal flora, digestions and detoxifications of exogenous substrate and metabolites. That is very important because most of bacteria present in gastrointestinal tract, it's help to digestions, it's help to remove toxins by formation of feces, and it's as a result of this to remove toxic products from human organisms. The next function, immunizations of human body, present of normal flora on site of human organisms, stimulate work of immune systems. Its immune systems always control that these bacteria don't spread in organ where it should be absent, as a blood and nerve system, maybe or another, in liver or kidney. And it's roles of immune system, and present of normal flora always stimulations and to work our immunity. And other functions. Some of bacteria produce vitamins which need for human organisms. And some of these vitamins don't produce our cells. It's produced only by prokaryotic cells, by bacteria. It's a group of vitamins B, E, H, and K. Also, some of bacteria produce amino acid proteins, which also don't produce in human body or produce slowly. That's why it's very important, and it's a function usually of gastrointestinal normal flora. And this bacteria also participation in metabolism of bile acid, lipids, carbohydrates. It's a good function of normal flora. But also we should be set and for negative functions. Then some of bacteria which present in normal flora may be conditionally pathogens. What it means? If it's suppressed of immune systems, this by infective endogenous can cause diseases. As example, staphylococcus, which can cause purulent infections. It may be present as conditional pathogen in oral cavity, on the skin and can cause diseases. It's only one example. Today we also be talking about normal flora of oral cavity, which can cause pathological processes, caries, periodontitis, etc. That's why not only good, but also negative functions present. Then, we discussed generally function of normal flora, and also today we will more tightly be discussed normal flora of oral cavity. But these functions, yes, the same and for normal flora of oral cavity, because all of these are character and for these bacteria, for these biotopes. Then normal flora of mouth and teeth. Then normal flora of mouth, sufficiency, moisturing and abundant food supply facilitates growth of organism in mouth. That's why it can grow and multiply. However, continuous flow of saliva remove of microorganisms from the mouth to stomach, and that's why most of this to kill it by acidity of stomach. Also, desquamation, sleeve of the tissue using mouth and another mechanism decrease the number of microorganisms at mouth. But part of bacteria survive and continuous multiply. The main, as I said, it's Streptococcus, Neisseria species, Lactobacilli, Veillonella, and Actinomycetes. Really, a more 10 in 8 microorganisms present in oral cavity, about from 150 to 180 different species are related from oral cavity, but us in this slide only a main to show. If we talk about in percent, then the 85% of microflora in mouse and in healthy dense mouse, yes, it's a Streptococcus, Veillonella, gram-positive diphtheroids, and gram-negative anaerobic rods. 5-7% genus Neisseria, about 2% Lactobacillus, 1% Staphylococcus and another Micrococcus gram-positive cocci, 2% filamentous bacteria as Fusobacteria, Actinomycetes, and another it's maybe another type of bacteria, maybe fungi as Candida, sometimes protozoa as Entamoeba gingivalis, and some of viruses. Then, and number and compositions of species bacteria in oral cavity differ from the different stage of the life in humans. Then, primary, when it's newborns to born and sterile, but very fast. Neonates contains lactobacillus as preabdominal flora during the vagina delivery. Then later, infants acquire gram-positive cocci, such as Streptococcus and Staphylococcus, from the mother's skin during the breastfeeding. That is the first bacteria will start to colonize mouth in newborns. The later, when start to development of tooth, oral flora changes and start a more gram-positive bacteria to add obligatory with development of tooth anaerobic bacteria, and main of this is Streptococcus and Peptostreptococcus. and use to Streptococcus and Peptostreptococcus at Veillonella, Bacteroides and Treponema. And in old, due to the teeth lost, microflora changing from gram-positive more to gram-negative bacilli, and as example, Veillonella. Then, compositions and amount of bacteria to change and depend of the stage of life, depend of the ages of human. You should be know that in oral cavity constant present concurrence between immune system, between another physiological mechanism of human organisms and bacteria. The oral cavity is one of the complex heterogeneous microbial habitat. Saliva contains microbial nutrients, but is not an especially good growth media because in nutrients present in low concentrations. Another in saliva present antimicrobial substances. The more famous is lysozyme, which cleavage glycoside linkage in peptidoglycan of bacteria, and as a result, lysozyme have bactericidal effect because it's lysis of cell wall of bacteria. Another enzymes which also present in saliva is lactoperoxidase. It's enzymes which present in milk and saliva and can kill bacteria which single oxidation in generate. That's why always present, yes, as concurrence between antibacterial substances which can be destroyed bacteria and cell debris provide high concentration of nutrients. These are present near the surface such as the teeth and gums, creating favorable conditions for extensive local microbial growth, tissues damaging and causes diseases. And another side of this problem, then different site of oral cavity contain different type of bacteria. It means and different number and different species. Then, if we talk about hard palate, then most of Streptococcus bacteria, bacteria from family Pasteurellacea, Veillonella, Prevotella, Lactobacillus, Gymella present in hard palate. On buccal mucosa, and most of Streptococcus and also Pasteurellacea. On tongue present, on tongue dorsum present Streptococcus, Veillonella, Prevotella, Pasteurellacea, Actinomycetes, Fusobacterium, Neisseria. In throat to Streptococcus at Veillonella, Prevotella, also Pasteurellacea, Actinomycetes, Fusobacterium, Lachnospiraceae. In saliva, Prevotella, Streptococcus, Veillonella, Pasteurellacea. Palatine tonsils, Streptococcus. Streptococcus villanella, Prevotella, Fusobacterium. Another word, look at these attentions, and maybe yes, as you understand, genus and species which will be studied. It's a Streptococcus. I always said it's a main microorganism which should be known stomatology or dentists. But if we talk about microflora of the tooth, then it's also be different. Some give out plaque formation by Streptococcus, Fusobacterium, Capnocytophaga, Prevotella, Carinobacterium, family Pasteurulaceae. Supergingival plaque formations, same Streptococcus, Capnocytophaga, Carinobacteria, but also to Neisseria and Fusobacteria. In keratinized gingiva also may be present Streptococcus and bacteria from family Pasteurulaceae. That's why in different site of oral cavity, different some of same species, genus and species, but another differ. And if we talk about amount, it's also different. If we talk about growth and multiplication, bacteria in oral cavity, then as another bacteria, this bacteria can grow the first as a suspension, but it usually may be present in this state in saliva. But if we talk about surface as tongues and in first as teeth, then bacteria from suspension, the first formation, colonies, and then formations biofilm or plaque. And it's shown on this picture on the first, yes, suspension, that is reversible adhesions, then it's colonizing the surface, formation colonies, then co-adhesions, bacteria can together multiply one to another, continuous multiply, to connect with different proteins, matrix, and formate plaque or biofilms on the surface of the teeth, on the surface of the gums or tongues or another. And today we discuss how it's step-by-step formations biofilm in oral cavity. When the teeth appear, balance of microflora shift toward anaerobes that are specifically adapted to growth on surface of the teeth and its gingival crevices. Bacterial colonizations of tooth surface begins with attachment of single bacteria cells, as I said. When the freshly cleaned tooth surface, acid glycoproteins from the saliva form these organic films, several micrometers thick. These films act as attachment site for bacterial microcolonies. And the first bacteria which start to sorptions on these organic films, it's a Streptococcus. This group of Streptococcus called oral Streptococcus. This Streptococcus sanguis, sorbineus, Streptococcus mutans, mites. And it's a first colonized glycoprotein film. Result of growth of these bacteria It's a formation of bacterial layers which called dental plaque. The next can be sorptions filamentous anaerobic bacteria as Fusobacterium. These filamentous bacteria embed the matrix formed by Streptococcus and extend perpendicular to the tooth surface, making ever thicker bacterial layers. Then later can be at Spirochetas as Borrelia species, gram-positive rods and gram-negative cocci. These bacterial congregations between one to another, and the formation of plaque can be continuous as a heavy plaque. When it's to add filamentous obligatory anaerobic organisms such as actinomycetes. And as a result of these formations, dental plaque can be considered as mixed culture biofilm that consists of a relatively thick layer of bacteria from several different genera, and also as well as accumulated bacterial products. The anaerobic nature of the oral flora may seem suppressing considering the intake of oxygen through the mouth. Result of this, we add this biofilm or plaque. It's a tooth surface, pellicula, acquired pellicula, yes, as I said, it's a peptidous glycan layers. The first, Streptococcus, which sorptions on the surface, Sangius, Streptococcus mitis salivarius. Then to add another bacteria, maybe Capnocytophaga, Haemophilus. Then it has gram-negative Veillonella, gram-negative cocci, Echinella, Prevotella, maybe. Then Fusobacterium as a binding bacteria which binding early colonizators with late colonizators. And after Fusobacteria can be at Aggregatobacter, Prevotella, Spirochetas as Borrelia and Treponema, Porphyromonas, and in other species. Let a more detailed as formations biofilm on delta surface. During the several hours, if it's, yes, for example, after professional cleaning of professional dense cleaning of tooth, during the several hours and until 2 days, start to formate biofilms. And as I said, the first start to sorptions, Streptococcus. Streptococcus aureus, mitis, gardoni, sangiu start to sorptions on the surface of glycoproteins films. These bacteria have different, very many mechanisms which help to sorptions and multiply in these conditions. And it's a first primary colonizer of biofilms. After this, start to sorptions actinomycetes, maybe Israeli, viscosus, Lundi, Haemophilus, as for example Haemophilus parainfluenza, Veillonella, maybe Echinella, and its attachment on the surface of Streptococcus, co-aggregations between with Streptococcus from one to another, maybe, for example, as on this to show how Veillonella can be aggregated with actinomycetes. That's why on pelliculus recognize salivary receptors and specifically bind them to help to adhesion proteins contribute to the attachment of following colonizators. It's an early colonizers or colonizator. Then intermediate colonizators, it's a Fusobacterium nucleatum. Very important because this intermediate colonizator binding the first layers, early colonizator and late colonizator. Fusobacterium gram-negative rods, filamentous, and it's helped to aggregate early and late colonizator. Later, on the surface of Fusobacterium start to options Treponema, Spirochetus, yes, it's maybe Borrelia, Treponema denticola, Parfenramonas gingivalis, maybe Aggregatobacter also to add, Actinomycetum comitans, Veillonella also maybe play roles as a late colonizator. Distinguished features of late colonizators, that is, cannot aggregate between very often, and most of it aggregate only with Fusobacteria. We discuss how to grow bacteria on the surface of the tooth of in oral cavity, how to formation biofilm, which are constant and always present. As I said, after several hours, start to formation biofilm after maybe professional cleaning, dental professional cleaning of teeth. But bacteria can to act on the structure of the tooth. Then how is to act and what maybe happens if it's a compensation mechanism to changes. We discuss now. Then, tooth enamel consists of mineralized matrix crystals of apatite, calcium phosphate. Yes, they are orientated form of enamel prisms, as which formate as its torpid, structure of dentines. Yes, that is enamel prism retains the shape of amyloblasts, that is synthesis of them. Normally, tooth enamel is a dynamic equilibrium between process of demineralizations and remineralizations. Demineralizations is due to the ions, hydrogens, organic acid of microbial regions. Destructions of enamel is accelerate at its pH below 5. Then, if it starts to decrease of pH less than 5, enhance risk of destroyed of enamel. And bacteria can initiate this process, can to decrease pH in oral cavity and as a result can initiate process of the caries. Then let's discuss what is this caries and what role to play bacteria in this process. Dental caries is process of demineralizations and softening of the hard tissues in tooth with the formation of cavities. What factors to enhance the development of caries? Start from the local factors. The first, presence of the plaque, place of accumulation, cariogenic bacteria, violation of the composition, properties and pH of saliva. Yes, it depends what pH of saliva. In aphids, present factors as lysozyme, peroxidase, etc., or maybe lack of these protective factors. Features of the structure of teeth. Present carbohydrates reduces of the food. It's a local factors. It's factors we checked in oral cavity, but also important general factors. General state of human organism. From general, it may be systemic diseases of the body, taking medications, state of immunity, genetic predispositions, and stress. All of these factors act on dynamic of biofilms or plaque on the teeth, and also it acts as a local factor and can decrease or increase risk of formation caries. Let's discuss what are first cariogenic bacteria, what are more important for formations caries. And the first is, as you understand, it's a streptococcus, a first adhesive bacteria which start to formate biofilm on the surface of the teeth. It's Streptococcus mutans, sanguis, metis, salivarius. From 30 to 60% of oral microflora. Then, let's characterize of these bacteria as a main caisogenic bacteria. It's alpha-hemolytic Streptococcus, gram-positive cocci arrangement in chain. It causes incomplete alpha hemolysis. It means formation green zone on the blood media, not light zone. These bacteria don't react with lens-filled antiserums, as for example in other pathogenic streptococcus as Streptococcus pneumonia, galactia. And it's differentiated by fermentation carbohydrates and production H2O2. The main role in pathology is the damaging of tooth tissues and periodontal diseases. Acidifies the medium, loss of the calcium in enamel. Synthesis polysaccharide as dextrans, insoluble a lot of dental plaques, formation matrix of dental plaque, and soluble glucan and levans. It's a source of formations acid and low to pH. And about these bacteria, about pathogenic factors and pathogenic properties of these bacteria, we will talk in more detail. Then, cariogenicity and virulence factors of Streptococcus mutans as the main groups of cariogenic bacteria. The first virulence factors, adhesins. It's adhesive proteins B1 and 2. Localization on cell wall of bacteria and help to adhesions, binding of saliva covered surface with teeth. its ability to give to sorptions and binding with surface of the tooth. The next factor, glycosyl transferase, different type, glycosyl transferase B, C, and D, are secreted and sometimes may be associated with cell surface. Formation of alpha-1,3 alpha-1,6 bound glucose polymers from sucrose and its help to adhesion and formation of biofilms. Next virulence factors, glucan binding proteins. Several types for different bacteria, glucan binding proteins A, B, and C. Localizations on cell wall, on cell surface. Binding glucans formed by glycosyl transferase, adhesion to the surface of the teeth, the formation of biofilms. The next virulence factors, fructosyl transferase, secreting enzymes, formation of beta-2, 1, beta-2, 6 bound fructose polymers from the sucrose, intracellular carbohydrate stores, and as a result, maybe yes, we always said for children, don't eat in a sweets because it's maybe yes to destroy it of your teeth, but you as stomatology should be known that not always present exogenous sucrose initiate process of caries because this bacteria can accumulate to different type of carbohydrates as a stores, and as a result, Streptococcus always have products to destroy it and to produce acid to lower of pH and to initiate destroy of enamel. The next virulence factors, fructanase, secreted or fixed in maybe secreted, maybe fixed in cell wall. The function of its hydrolyzing of fructans formed with participation of fructosyl transferase contributes to the acidifications of the environment, as a result also to low of pH. The next virulence factors, dextranase, secreted enzymes, breakdown of alpha-1,6 bound glucans, leading to increased release of the glucose, which broke down to form acid. And we continue, it's not another factor. Intracellular polysaccharide, formation inside the cells, glycogen-like polymers of glucose. When exogenous glucose is depleted, it's used as supply of polysaccharide, causes acidifications of environment. As I said, Streptococcus always formation storage of polysaccharide as source of energy, and as a result, always formation acid during the destroy of these carbohydrates. Another virulence factors, acidity and adaptation to acidity environments. For Streptococcus, a comfortable this low pH, because usually for bacteria comfortable neutral pH, but Streptococcus very good survive in this low pH. Then it have FATP other. It's a multi-factor process allow bacteria to increase resistance and grow more efficiently to a low pH values. We discuss the first or primary bacteria which are important for formations biofilms, which play a main role in formation of caries. We discuss pathogenic factors of these bacteria, but not only Streptococcus play a role to formations, caries, and formation cavity in a male. The next bacteria is Lactobacillus or Lactobacilli. In English, Lactobacillus in Latin, Lactobacillus acidophilus, Lactobacillus fermentum, Lactobacillus brevis. It's gram-positive rods, anaerobic bacteria, and properties of these bacteria decompose carbohydrates as result formations lactic acid and as result to low of pH. That's why participation in pathology, it's acidification of environment and as a result initiate lack of calciums and to destroy it in the male. The next bacteria, we talk about this, Actinomycetes. Actinomycetes israeli or viscosus as a famous species. Very interesting bacteria. The name of these bacteria says that primary thinking, that is not bacteria, Actinomycetes. Yes, it's a fungi. Thinking the primary, that is maybe fungi. Why? Because these filamentous or branching maybe multiply by budding, by spore and divisions. But later when to start to study structure, cell wall compositions, and to understand that is really bacteria. That's why now thinking that it's gram-positive, but it's maybe variable, maybe sometimes staining as gram-negative. Gram-branching bacteria, 0.212 micrometers in length of 60 micrometers or 600 micrometers. They found in dental plaque, as I said, participation and development of dental caries, but that is also important for pathology of gingiva pockets and periodontal diseases. How? It's also formed acid and as a result of low of pH. Except bacteria, also we can add to cariesogenic flora fungi as a candida. More famous species, Candida albicans. It's a yeast-like fungi of aerobes or facultative anaerobes. It's very good growth on low pH and 5. Synthesize vitamins, growth factors for lactobacilli, and can acidify the environment. That's why it's not direct, but it's some of direct acidify the environment and indirectly stimulation growth of lactobacilli. Except participations in indirectly to caries, it's also play roles to development of stomatitis, especially with dental prosthesis. And the next, Streptococcus. Streptococcus asaccharoliticus. Gram-positive cocci, sometimes arrangement in chain, and as its name, asaccharolyticus, and it said that it's to don't have saccharolytic activity or weakness saccharolytic activities, but very active against proteins or peptones, actively decompose peptones and amino acid. As a result, participation in pathology together with fusobacteria and spirochetes, they participate in development of caries, pulpits, periodontitis, and abscesses the maxillofacial region. The next bacteria, today we talk very many times about this bacteria, fusobacteria, as intermediate colonizator. It's gram-negative rods with thin ends. Yes, it's specifically structure. Gram-negative fusiform rods. Obligate anaerobes, don't formate spores, but formate lactic, then it's also too low of pH. Together with spirochetes and bacteroid can cause one of maybe not often but very difficult to treat in diseases as cause ulcerative necrotic Vincent stomatitis. Species Fusobacterium periodonticum, Fusobacterium nucleatum. But as I said, formation of plaque or biofilms on the surface of the teeth, it's a dynamic process, and if it's a health oral cavity, in this plaque present not only cariosogenic bacteria, also present carious-limiting microflora. And the more famous carious-limiting microflora is Veillonella. Veillonella gram-negative diplococci, it means gram-negative coccus, which arrange in pair, strict anaerobes, and it's to limit formations or to inhibit cariosogenic bacteria by decomposition of acid. Decompose, lactate, pyruvate, acetate, and another organic acid to produce carbon dioxide and H2, water. Then, participation in pathology, it have anti-carious effect because they compost lactic acid and another acid. As a result, increase of pH and environmental and process of demineralizations and remineralization is stabilizations. Then we discuss with you very short normal flora of the main bacteria from normal flora of oral cavity, especially from bacteria which formation teeth plaque, and we talk about roles of bacteria in caries, and maybe I need to talk about another diseases in oral cavity. As periodontium, soft tissues, oral cavity diseases, and maxillofacial regions. Then, bacteria or microorganisms, causative agents of infection diseases of periodontium, soft tissues, and maxillofacial region. Another word, odontogenic infections. If we look on this slide, then maybe we see different variation of odontogenic infections. One of these, it's a problem of dentist, of stomatology. Another, it may be problem in other doctors and be treated with another specialist. Then, the first, we talk about diseases which should be treated in stomatology doctors. It's a pulpits, infections and spread bacteria in pulp cavity. It's periodontitis, bacteria cause inflammatory and destruction properties in bone near the teeth. Periodontitis, inflammatory of tissues near the teeth. Also maybe abscess phlegmos of soft tissues of face and neck. But also bacteria which present in teeth, in dental, in pulp, maybe in periodontitis, can cause another type of diseases. If it's spread in blood and spread in another organ and tissues, it can cause sepsis, can cause sinusitis, can cause osteomyelitis. That's why bacteria which can present in oral cavity can be the reason of another infection diseases. But today we talk only about problem of dentists. Pulpits. An inflammation of soft tissue of the tooth pulp as a result of caries process. The main causative agents. Maybe you understand this, it's streptococcus. We discussed, yes, it's a gram-positive coccinine chain, facultative anaerobes. Except that it's acidified environment, it also can cause purulent inflammatory diseases, initiate purulent inflammations. The next bacteria as cariosogenic bacteria, lactobacilli, Lactobacillus brevis, acidophilus fermentum, gram-positive rods with different lengths, which not format spores, facultative anaerobes or microaerophilic. Also, yes, acidifications and cause inflammatory diseases and pulp. If it's don't treat in carrier, that is spread in cause pulpits. The next bacteria, which we don't discuss, yes, previously, it's a bacteroids, Bacteroid fragilis, gingivalis. It's a gram-positive rods, capsulated, don't form spores, as on this picture you can see, but it's not gram staining, it's low by gram staining. Microaerophilic and obligate anaerobes, ferment sugars to gas, peptones and amino acid, smelling. As a result, yes, it's a smell not from the oral cavity, it's a result of destroyed by these bacteria, peptones. Causes purulent necrotic diseases. Why this bacteria cause this problem? Because it produces viral factors as enzymes, collagenase, DNase, heparinase, fibrinolysins, beta-lactamase. Collagenase destroys collagens, DNase destroys of DNA of cells, destroys heparins as heparinase, fibrinolysins can destroy fibrins and which give ability to spread this bacteria in tissue. Beta-lactamase give ability to multiply this bacteria in presence of the beta-lactam antibacterial drug. It's antibiotics means. Some of bacteroids release endotoxins as leukocidins, fatty acids, and presence of polysaccharide capsules which protect these bacteria from phagocytosis give ability to multiply these bacteria in pulpa, maybe in other soft tissues. It's a main pathogenic factors of bacteroids. Another another bacteria which can initiate another type of pulpites, it's a purulent process. It's a alpha hemolytic streptococcus and beta hemolytic streptococcus. About streptococcus we know all. Alpha hemolytic, as I said you, yes, it's formation green zone of hemolysis, incomplete hemolysis. But also if we talk about pulpites, may add beta hemolytic streptococcus, as for example Streptococcus pyogenes, which formation completely destroyed erythrocyte and formation beta hemolysis late zone. The role of this is acidifications of the environmental synthesis polysaccharide, insoluble extract, dextrans, and as a result give ability to formations plaque and initiate inflammatory process. Also, if we talk about purulent process, to maybe add Staphylococcus aureus. You all know that is gram-positive coccyte, arrangement from bunches or clusters like grape. About 25–40% of populations permanent carriers of this bacterium. That's why this bacteria persistent on the skin, on mucous membrane, upper respiratory tract, oral cavity, and during the destroyed or change in normal process in health organism, it can initiate different purulent diseases and also in oral cavity. Staphylococcus aureus have different virulence factors. It's produced different type of membranotoxins which destroy different type of the cells. It's alpha, beta, gamma, delta toxins, or leukocidins which destroy erythrocyte, leukocyte, and another. Staphylococcus produce toxic shock syndrome toxins, which spread in blood and can cause, if it's spread in blood, it can cause shock syndrome. Some of Staphylococcus produce enterotoxins, which can cause food poison. Another type of strain Staphylococcus can be produce exfoliation toxins, which destroy desmosomes between cells and cause desquamations. Also, Staphylococcus produce different type of aggressive enzymes as coagulase, lipase, hemolysins, also formations alpha hemolysis, as another as a Streptococcus. This bacteria formation a capsule which protect against phagocytosis. Protein A, which inhibit also phagocytosis, protect against immune systems. Tehoic acid, which help to adhesion these bacteria on the surface of tissue. Fat acid, which can stimulations of inflammatory process. And on this picture also to show production of coagulase, test for production of coagulase. Coagulase positive Staphylococcus produce these enzymes which coagulase a plasma in blood. And it's a coagulase negative, yes, remains a liquid. Wide importance for Staphylococcus in human organism, because during the local infections and in oral cavities very often, these bacteria produce these enzymes, coagulase, and plasma clot, which formation surrounds the place of multiplications. Bacteria don't give ability cells of immune system, another element of immune system, to spread to place of multiplication of this bacteria. As a result, by these enzymes, bacteria protects against action of immune systems. And if we talk about another type of pulpids, putrefactive process. That's why to add made to bacteria, add a more anaerobic bacteria as bacteroids, as proteus, Streptococcus, Veillonella, and Clostridia. And most of this we talk, yes, bacteroids we characterize, Streptococcus also, Clostridia we study, yes, it's Clostridium novi histolyticum, which can cause anaerobic wound infections. Veillonella, it's also we characterize. But if these bacteria spread in pulps, it can cause putrefacitive process. But except pulpids, also maybe bacteria which present on the surface of the teeth can initiate inflammation in soft and hard tissues around the teeth, with impaired attachment of collagens to the teeth. As a result, bacteria can cause periodontitis. How bacteria to spread in periodontium? One way, through the teeth canal from inflammatory pulp, and another, less often, penetrate between wall of alveoli and root of the teeth with periodontopathies, or with hematogenous drift of the infection. But one or another way, bacteria spread in periodontium and cause inflammation process. What consequences of this inflammation process? Microorganisms can destroy connective tissues with enzymes, collagenase, hyaluronidase, neuraminidase, and induce inflammations. Inflammations of the mucosal membrane of the gums can lead to the loss of bone tissues of the alveolar process, with subsequent loss of the teeth. Then, don't treating long-time periodontitis can cause loss of the teeth. What bacteria can initiate this process? Streptococcus, Staphylococcus, Lactobacillus, same, yes, name, but also to add Carinobacteria, yeast-like fungi, Veillonella bacteroides. And more often, during the periodontitis, associated association in adults is gram-negative anaerobes, Pheromonas gingivales, Bacteroides furcintus, Prevotella melaninogenica, facultative anaerobic rods as Actinobacillus actinomycetem comitans and Treponema denticolor. Associated in adolescents, its facultative anaerobic bacilli as Actinobacillus actinomycetem comitans and different species as a capnocytophaga. Let us short characterize some of causative agents. The first we talk about Parpheromonas gingivalis. It's a short, non-motile gram-negative rods, about 1,3 and 0.5–0.8 micrometers, small, gram-negative rods. Distinguished features than this bacteria on the media with blood formation brown-black pigment colonies. And another distinguished feature is growth only in blood because it's need hemines and vitamin K. That's why we can see, yes, this is colonies, black colonies on the blood agar. If we talk about virulence factor, that is adhesion and invasive, with participation of fibrous hemagglutinins, proteins of the outer membrane, and ability to destroy fibrinogens. Also, these bacteria secrete collagenase, involved in dentin damage, and also agglutinase erythrocytes. The next causative agents, Prevotella melaninogenica. Prevotella, polymorphic, non-motile gram-negative rods. Don't form as spores, but have a capsule. Obligate anaerobes. Fermentate carbohydrates or sometimes particular fermentate. Prevotella melaninogenica secreted virulence factors as protease, phospholipase A, neuraminidase, which give ability to these bacteria integrate in epithelial cells, and which as a result it can cause the death of these cells. Also, this Prevotella can cause release endotoxins, and then actions of these endotoxins is more intensive than lipopolysaccharide of bacteroids. Another causative agents which very important play very important roles of periodontitis, it's Aggregatobacter, or previously it's called Actinobacillus, but now Aggregatobacter actinomycetem comitans. Distinguished features that is very often aggregations and with actinomycetes and present and other lesions also together with actinomycetes. That is named actinomycetem comitans. It's gram-negative rods. From causative agents, we can set about fibrous, which help to adhesions, matrix glycoproteins, which also play roles of adhesive proteins, and to also describe bone resorptive activity. Also, bacteria produce protease which break down collagen, fibrinolactins, immunoglobulin G and M. As a result, it's to destroy it of immune mechanism, and it gives ability to continuous multiply. Bacteria produce exotoxins. One of these leukotoxins pore-forming toxins, formation pore in membrane, often infecting cells, and other holotoxins contributes to the swelling and stopping the life cycle of eukaryotic cells. Another also destroyed eukaryotic cells. And as these gram-negative bacteria, that's why it's release endotoxins, and this is properties of these endotoxins described as bone resorptions, platelet aggregations, and necrosis. That's why it's a highly aggressive bacteria, especially in organisms very suppressed of immune systems. And another bacteria, as Treponema denticola, spirochetes, gram-negative, but not usually gram-staining, as in most of spirochetes. Bacteria adherent on human gingival fibroblasts under aerobic and anaerobic conditions due to the proteins of the outer membrane, which has a cytotoxic effect. Invasion associated with motility of these bacteria and production of protease. Action of protease, hydrolysis of fibrinogens, transferrins, serum albumins, and immunoglobulins. The next, Capnocytophaga species of different species of Capnocytophaga. These bacteria long, thin, spindle-shaped gram-negative rods, facultative anaerobes, grow better in atmosphere rich with carbon dioxide. The more famous species which can cause periodontitis, Capnocytophaga orchacea, Capnocytophaga gingivalis, and Capnocytophaga sputigen. Capnocytophaga usually is part of normal flora of oral cavity, but during the suppressed of immune system, especially for patients with leukemia, Capnocytophaga can cause sepsis. And another, but very often suppressed of defense mechanism of human body, it's a in adolescence period. That's why in this period, very often this bacteria initiate periodontitis for adolescent or for teenage. That's all. Then our course of lectures to finished. Goodbye.
Ножи.